How do we put it on the projector? Is it on? Is it just full screen? No? No? It's just a PDF.
I don't know what you got.
I didn't touch anything. There we go.
Okay, so we should get started. Our last speaker of the COSI and the last speaker before the keynote is Natalie Sauerwald from Carnegie Mellon. And I should remind people that this is a proceeding presentation. So,
All right. Thank you so much for the opportunity to talk about my work in comparing topological domains across human cell types. And thank you all so much for sticking around and coming back for my talk. You're almost at the end. So as many of you probably already know, chromosome structure is implicated in several really core genomic functions. We heard a talk earlier about replication timing domains, and structural domains have been shown to correlate really highly with replication timing and to be really implicated in the coordination of replication. And chromosome structure is also really strongly associated with gene regulation, with gene expression levels and with the activation or repression of genes. So related to that, chromosome structure is not surprisingly also implicated in several human diseases. So many cancer types have been shown to have mutations that affect the 3D structure of the chromosomes. And it's also been shown that you can actually activate proto-oncogenes simply by changing the 3D structure around them. There's also several human limb malformations that have been shown to be caused by relatively minor structural changes around the genes that control human limb formation during development. And several really severe brain anomalies that are also have been shown to be due to several minor structural alterations. So when we talk about chromosome structure, many of you may be very familiar with this already, especially if you attended all the RegSys talks, but for those who didn't, what we talk about with genome structure is mostly these structures called topologically associating domains. So if you sort of zoom in on a chromosome in the nucleus, you can see these structures of really more tightly interacting chromatin. So the definition I'm going to use here is regions that are generally under a megabase in length that interact much more significantly within the domain than across domains. And so the way we measure chromosome structure, what happened to that slide? Well, my high C slide is gone. But the way we measure chromosome structure is with a matrix, a high C matrix, where each row and column represents a segment of the chromosome, and each entry represents an interaction between those two segments of the chromosome. So the way those look in, the way TADs show up in a high C matrix is these darker, more highly interacting triangles along the diagonal of that matrix. So you can sort of see here, if you picture this matrix, like I said, each row and column represents a segment of the chromosome, and these high interactions can sort of cluster in these TADs. So a sort of natural question that arises when we talk about describing TAD structure is, how does it vary across different contexts? So how similar is TAD structure across chromosomes, across cell types, across cancer and non-cancer cells? And then, oh God, like half my slides are gone. All right, so how we could measure structural variability. Sure. I'm not sure. I just went through this PDF and it was, I'm not sure. Well, I don't have my computer with me. So hopefully the rest of them will be here. So people have compared high C matrices directly. Behind this box, there was supposed to be two high C maps. But the problem with that is that high-C processing and the coverage differences can really mask underlying structural similarities, and especially when you're trying to compare things across different data sets from different labs, it can be really difficult to overcome these artifacts from the protocols. And so our solution to that is to compare TAD sets directly rather than the full high-C matrices. And the other way that people have measured structural variability in the past is these Venn diagrams you can see on the bottom. And all those do is count the overlap or the number of boundaries that were common between two cell types or two species. But these Venn diagrams really don't identify conserved structures, right? They just tell you the number of overlapping boundaries. And they don't associate this, whatever similarity they capture, with any sort of functionality. So what we'd like to do here is identify regions of locally similar structures. So if we could do this, a deeper understanding of structural variability could lead to greater evidence for 3D gene regulatory mechanisms. We could identify some cell type or maybe condition-specific structures, get some deeper insight into the relationship of chromosome structure with disease on a more global scale than we have right now, as well as an estimate of the expected structural variance across different individuals or cell types or contexts. So when you get a new high-C map and you say that the structure looks really weird, you can tell if it's a true outlier or if it fits within the sort of expected variance of the distribution. So our goal here is to identify regions of structural similarity between two TAD sets. The inputs here, there was another figure in the middle there. The input here is these two TAD sets you can see that are represented as just two sort of lengths of triangles. If you outline the TADs along the diagonal, they look like a set of triangles, so I'm going to represent them that way here. And we just like to identify where those are structurally similar. So our method, here's a quick overview. Again, it takes two TAD sets as inputs. You can use whatever TAD finder you want. And then we look at the distance between every possible subinterval of these TAD sets. So if you cut it up at every single boundary point and compute the distance between the TAD sets in that subinterval, all those red dots represent distances that were computed between subintervals of these TAD sets. And since there's a lot of possible subintervals, we came up with a dynamic programming algorithm to make this computation more efficient. And then we run a statistical significance test to keep only the ones that seem really meaningfully similar to each other. And then to refine the results a little further, we have a heuristic that we call dominating intervals, and the output is, again, regions where the set seems structurally similar. So I'm not going to go too far into the technical details, but a couple things to mention. The distance metric we're using is variation of information, which is a general metric from information theory that computes the distance between any two clusterings. So we're considering basically our TAD sets as clusterings, where each cluster is the set of high-C bins that are in the same TAD. And then the intuition of this metric is it quantifies the information, so this is computer science information in units of bits, that each clustering provides about the other. So here's the equation for VI. It's just the sum of the conditional entropies, and we normalize, so it's between 0 and 1. And the conditional entropies quantify essentially the information or the bits you need to describe one clustering given that you know the other. So I mentioned our dynamic programming to make this more efficient. And the idea here is that if we are given two subintervals, so we know the VI between C and C prime, and we know the VI between D and D prime, and we want to compute the VI between this whole thing. Rather than recomputing all of those probabilities and logs, we can simply take a linear combination of the VIs of the subintervals. So this is the simple case. It's a little more complicated if you, let's say CD didn't actually have a boundary there, but we artificially chopped it off. You can still do dynamic programming. The details of that are in the paper. It's just a little too ugly for the slides. And implementing all of this empirically reduced our computation time by almost 60%. So the other technical detail is our statistical significance test, which is just a permutation test. We fix one TAD set. So for every single subinterval, we fix one TAD set and then randomly permute the other set n times, computing the distance each time. And that gives you these two vectors of distance values, and you just want to count how many of those distances are less than our original. So if we randomly permute the TAD sets, how many times did we end up randomly getting a better match than we had originally? And then since we are testing lots and lots of possible subintervals, we use Benjamini-Hochberg to correct for multiple hypothesis issues. And our dominating interval heuristic comes after the statistical significance. So looking just at the set of statistically significant intervals, we remove any subinterval that has, we remove anything that has a subinterval with a lower distance. So if you can get a better match by just taking a smaller interval, we'll just keep the smaller one. And then we still had some that started or ended at the same place, so we just kept the longest of those. So here's an example of what the output looks like. In the method overview is a toy example. It doesn't look that great, but these are real TAD sets. So in green, the triangles on top are the TADs from A549 cells, and in blue on bottom, we have TADs from KBM7 cells. And then these red sort of boxes here represent the regions that our method said were structurally similar between these two TAD sets. So this one in here is a nice example. You can see that the TADs line up almost perfectly in this region. It's just a good demonstration of what we were trying to find with this method. So using this, we ran a study of 23 human high-C samples from these 4 different labs of various coverages and cell types. And we did all possible pairwise comparisons of these 23 samples, which is 253 pairwise comparisons. And we wanted to look at, like I mentioned earlier, the variability across chromosomes, across cell types, and between cancer and non-cancer. So I'm going to go through those 3 categories here. So first, looking at chromosomes, the x-axis here is the genomic position along the chromosome, so how far along the chromosome are you? And the y-axis is the fraction of structurally similar pairs. So a high value here indicates that that region was really similar structurally across a lot of our comparisons, and a low value indicates that it was like poorly conserved. It didn't appear to be similar across many of our comparisons. So you can see a whole lot of structural variability both within individual chromosomes and between multiple chromosomes. Just so you think I'm not cherry picking, here's the results on all 22 autosomal chromosomes. So these are basically just box plots of the distribution I showed in the last slide. So again, the y-axis is the fraction of structurally similar pairs, and we have a box plot for each autosomal chromosome. And you can see that there's a huge amount of variability. A lot of the chromosomes have values that go from 0 to 1, so there are both bins that were not similar across any of our 253 comparisons, and ones in which were similar in all of them. There's also some variability between the chromosomes, especially obvious in chromosomes 1 and 3 here, which for some reason chromosome 1 shows a significantly lower distribution overall, and chromosome 3 is much higher. So we also talked about looking at comparisons between cell types. So the method gives us sort of a natural measure of global similarity, which is just computing what % of the genome is covered by these structurally similar intervals. And we computed that for each one of our 253 comparisons and put it in this heat map. We don't see a whole lot of structure here, but what we did find is that these cell types at the bottom that show very little similarity overall with anything else tend to be basically just the lower coverage, lower quality data sets. We found similarity values between about 30% and 60%, which is consistent with the Venn diagrams I showed earlier. So we also were kind of happy to see a lack of structure in this because we were worried about just capturing batch effects. So if you kind of color these by what data set they came from, we don't see blocks of these data sets aligning with each other. So now we've talked about variability in chromosomes, in cell types, and we're going to talk about cancer now. So this plot's a little complicated, so bear with me for a second. Again, we have 22 box plots for each chromosome, but now the y-axis is a difference in % similarity between the pairs of normal cells and the pairs of cancer cells. So values above 0 here represent where there was more structural similarity among our normal cell types, and values below 0 represent where there's more structural similarity among cancer cell types. So interestingly, there's not a huge pattern of like global conservation or global disruption in cancer, 10 of these 22 chromosomes have averages below 0. But if you look at chromosomes that contain pan-cancer genes, so these red stars represent the top 10 most commonly mutated pan-cancer genes from a Nature study a few years ago. And 9 out of the 10 of those lie on chromosomes that are much more structurally conserved among normal cell types. So if we look a little more closely at that, this plot shows the values at those 10 locations. So these are these top 10 cancer genes that I'm sure we're all very familiar with, things like TP53. And the purple bars here are the structural similarity, fraction of structural similarity among pairs of normal cells. And the blue bars represent the structural similarity among cancer cells. So 9 out of the 10 of these were much more structurally conserved among normal cells. So this might point to the 3D disruption that happens with these cancer mutations. But we need to do a little more study with this. So to summarize, this is the only method so far that directly quantifies and compares TAD structure. We were able to do some local and global structural comparisons through 253 pairs of human cell types across a bunch of different tissues and cancer states. We saw a lot of structural variability within and across chromosomes and between cell types, and also observed higher structural variability among cancer cells at specifically pan-cancer gene locations. So this is in the Proceedings issue, Bioinformatics. Please take a look if you are interested, if you want more details. The code is also available on GitHub, so if you're doing any sort of experiments where you're trying to compare structure across any context, please feel free to use our software and let me know if there are any issues with it. And so I'd just like to acknowledge the Kingsford group and the fantastic people in it. We are looking for postdocs, so if you're interested in this or any of the other talks by Kingsford group members, let me know. I'd like to thank ISCB for the travel funding and all of our other funding sources. And I'll take questions now.
You basically first defined a metric for doing the comparison, and then you went and used it to sort of, find about a bunch of very nice results. My question is, have you gone back to look at reformulations of the basic low-level similarity metric now
like
that you have a set of cell type comparisons and so on and so forth to basically see how robust your results are to that metric per se?
You mean to the variation of information metric?
Correct. Yeah. There's many ways that one could compare TADs, and I'm sure that you hesitated a lot before coming up with that metric, which sounds sensible. But my question is, if you have an evaluation set, now that you have a bunch of cell types and so on and so forth, you could basically say, if I adjust my metric, which one is actually better reflecting of cell type hierarchies and so on and so forth?
Yeah, it's a great question. We have talked about different distance metrics. Like you said, there's a million possibilities for comparing clusterings.
Approximate value, yeah.
Yeah, approximately a million. So we were exploring that a little more now to see if there was a better one. VI has been used to compare TADs in the past, so it seemed reasonable. But hopefully those distance metrics should all be relatively consistent because they're all measuring a distance.
Of course.
But yeah, we're looking into it.
But I think it can give you also insights as to what should we actually be thinking about when we think about TAD conservation and similarities.
Yeah. Thank you.
I have a question. In your approach, you assumed that the TAD boundaries are fixed and you compare them to fixed structures. But in practice, often you can have some confidence or depth or significance for TAD boundaries. So could you somehow generalize your model to account for the fact that different TAD boundaries insulate to different levels, so to say?
Yeah, that's a really interesting question. That could relate to Manolis's question about distance metrics, that if you had a distance metric that could take into account a weight on each boundary or something, or a confidence level, you could incorporate that pretty straightforward into the model. But we haven't looked into it. It's really interesting.
So what type of features have you found correlating with the variability or lack thereof?
That's a great question. That is ongoing work. We're doing a lot more data analysis with a lot more TAD sets now to try to figure out. we expect to see some epigenomic, CTCF and the known histone markers, but we're looking to see if there's anything else that might be a novel relationship, but we're working on it.
I have a question for the cancer part. So you're saying that maybe the cancer genes may have a lower conjugation, which means they have some different kind of structure. So that means that you think, you just told CTCF, but CTCF is known to have kind of more cancer somatic mutation site inside its motif. So do you think this is one of the reasons?
It might be. It's certainly known that genetic mutations can cause structural, or can be related to structural disruptions. There's studies where they do CRISPR and mutate a region and then look at the structure and it is altered. So I'm not sure if it's related to binding proteins or things like CTCF, or if it's simply based on sequence alone, but they're all reasonable explanations.
And also, if you're using this kind of approach, then maybe you think you can find some kind of novel oncogenes?
Possibly, yeah. We've also been asked if we could predict genes that are cancer related through this. It's a little difficult because of the, oncogenes tend to be very cancer specific, and we don't quite have enough data to do cancer specific predictions. But possibly with more data, we can do more predictive work.
Thank you.
Thanks.